Chapter 15. Microbial Behavior as a Digestive Output. Microbial behavior in the gut reflects the condition of digestion rather than acting as an independent driver of symptoms. The microbial population responds to the substrates that reach it, and those substrates are determined by how effectively upstream processes reduce and absorb nutrients. When digestion proceeds in sequence, most proteins, fats, and carbohydrates are processed into absorbable forms before reaching the distal intestine. The microbial environment then operates on a limited and consistent substrate pool. When digestion is incomplete, larger fragments and unprocessed nutrients move downstream, expanding the available substrate and altering microbial activity. The composition of luminal contents defines microbial behavior. Bacteria metabolize what is present, not what was consumed. When proteins are fully reduced and absorbed, minimal protein substrate reaches the colon. When breakdown is incomplete, larger peptides and amino acids persist, providing material for microbial metabolism. The same applies to carbohydrates and fats. Undigested carbohydrates increase fermentable substrate, while improperly processed fats alter the physical environment in which microbes operate. The microbial response follows these changes directly. Fermentation is the primary expression of this response. When fermentable substrates increase, microbial activity accelerates, producing gases and metabolic byproducts that change the internal environment. This affects motility, pressure within the intestine, and the chemical composition of luminal contents. The system reacts to these changes through adjustments in transit speed and secretion, but these responses are driven by the altered substrate conditions created upstream. The distribution of substrates also influences where microbial activity is concentrated. When digestion is aligned, most substrates are absorbed earlier in the small intestine, and microbial activity remains more distal. When processing is incomplete, substrates appear earlier and in greater amounts, shifting microbial activity proximally. This changes the pattern of fermentation and alters how the intestine experiences and responds to microbial metabolism. Microbial populations adapt to the substrates they encounter. A consistent input produces a stable microbial pattern, while variability in substrate delivery leads to shifts in population behavior. These shifts are not random. They reflect changes in nutrient availability and the physical environment within the gut. When upstream digestion is stable, microbial activity remains predictable. When it is not, microbial behavior fluctuates in response to changing conditions. Immune interaction with the microbial environment is also influenced by digestion. The intestinal barrier is exposed to both microbial products and residual dietary substrates. When digestion is complete, exposure to larger, reactive molecules is limited. When it is not, these molecules interact with the epithelial surface alongside microbial byproducts, increasing signaling within the immune system. This interaction reflects the combined effect of digestion and microbial metabolism rather than the action of microbes alone. Microbial behavior therefore represents an output of the digestive system. It is shaped by the substrates delivered to it, the timing of that delivery, and the physical conditions within the intestine. When digestion is aligned, microbial activity operates within a stable framework. When it is not, microbial behavior shifts in response to increased and variable substrate availability, and these changes propagate through the rest of the digestive system. Fermentation from incomplete digestion. Fermentation increases when substrates that should have been reduced and absorbed earlier in the sequence reach regions of the intestine where microbial populations are prepared to metabolize them. This is not a random occurrence. It reflects a failure of upstream processing to convert macronutrients into absorbable forms. When proteins, carbohydrates, or fats remain partially intact, they become available to microbes as alternative substrates. The intensity and location of fermentation are determined by how much of this material escapes earlier stages and where it accumulates. Carbohydrates are the most immediate driver of fermentation because they can be rapidly metabolized when they are not fully reduced to simple sugars and absorbed. Larger carbohydrate fragments that persist in the lumen provide a continuous substrate for microbial activity. As these molecules are metabolized, gases and byproducts are generated, altering the internal environment. This change affects motility and pressure, creating a feedback loop where the system adjusts transit speed in response to the altered conditions. Protein fermentation follows a similar pattern but reflects a different type of upstream failure. When gastric and enzymatic processing do not fully reduce proteins to small peptides and amino acids, larger fragments remain available for microbial metabolism. These substrates are processed into compounds that change the chemical environment within the intestine. The presence of these compounds influences how the intestinal lining responds and how motility is regulated, contributing to variability in digestive behavior. Fat contributes indirectly to fermentation when its digestion is impaired. Poor emulsification and incomplete breakdown leave lipids in forms that alter the physical environment of the intestine. This can change how carbohydrates and proteins are processed and how substrates are distributed within the lumen. As a result, fermentation patterns shift, not because fats themselves are rapidly fermented, but because they influence the availability and distribution of other substrates. The location of fermentation reflects where substrates first exceed absorptive capacity. When digestion is aligned, most substrates are absorbed before reaching regions with dense microbial populations, and fermentation remains limited and distal. When upstream processing is incomplete, substrates appear earlier and in greater amounts, shifting fermentation proximally. This change increases the interaction between microbial activity and the upper sections of the intestine, where the system is less adapted to handle large amounts of fermentation. The consequences of increased fermentation extend beyond gas production. The byproducts of microbial metabolism alter the luminal environment, affecting pH, motility, and epithelial interaction. These changes influence how the intestine processes subsequent substrates, creating a cycle where incomplete digestion leads to fermentation, which then alters the conditions for further digestion. Fermentation from incomplete digestion, therefore, represents a downstream expression of upstream failure. It reflects the presence of substrates that were not reduced and absorbed in sequence. The intensity, location, and effects of fermentation are determined by how much of this material escapes earlier stages and how it interacts with the microbial environment that receives it. Substrate overload and gas production. Gas production increases when the amount of undigested substrate entering a region of the intestine exceeds the local capacity for absorption and controlled processing. This condition develops when upstream stages fail to reduce macronutrients into transportable forms, allowing larger fragments of carbohydrates and proteins to persist in the lumen. These fragments accumulate and become available to microbial populations that rapidly metabolize them. The result is not simply the presence of gas, but a shift in how the intestinal environment handles incoming material, driven by excess substrate rather than normal digestive flow. The threshold for overload is determined by the balance between delivery and processing capacity. When gastric emptying is regulated and enzymatic activity is aligned with substrate arrival, nutrients are reduced and absorbed before they accumulate. When this balance is disrupted, larger amounts of partially processed material enter the intestine in a shorter time frame. Enzymes cannot keep pace with the incoming load, and transport systems cannot absorb substrates quickly enough to clear them. The excess remains in the lumen, where microbial metabolism becomes the dominant pathway for its breakdown. Carbohydrates are the primary contributors to rapid gas production under these conditions because they are readily metabolized by microbes when not fully reduced to absorbable sugars. As microbial activity increases, gases are generated as byproducts of substrate breakdown. The rate of gas production reflects both the amount of substrate available and the speed at which microbes can process it. When substrate load is high, gas production rises quickly, increasing luminal pressure and altering the mechanical state of the intestine. Protein contributes to gas production when it is not fully reduced and absorbed earlier in the sequence. Larger peptide fragments that reach the distal intestine provide additional substrate for microbial metabolism, generating a different profile of byproducts that further alter the luminal environment. These compounds influence motility and epithelial interaction, compounding the effects of carbohydrate-driven fermentation and contributing to variability in digestive behavior. Fat influences substrate overload indirectly by shaping the conditions under which other macronutrients are processed. When lipid digestion is aligned, it slows gastric emptying and distributes substrate delivery over time, preventing sudden increases in load. When fat processing is impaired, this pacing function is reduced and larger amounts of partially processed material can enter the intestine more rapidly. This accelerates the transition from manageable substrate levels to overload, increasing the likelihood of fermentation-driven gas production. The mechanical effects of gas accumulation feed back into the digestive sequence. Increased pressure within the lumen alters motility patterns, changing how contents move and how long they remain in contact with absorptive surfaces. These changes can further reduce processing efficiency, allowing additional substrate to persist and reinforcing the cycle of overload and fermentation. Substrate overload and gas production therefore represent a quantitative failure within the digestive sequence. When the amount of unprocessed material exceeds the capacity of enzymatic and absorptive systems, microbial metabolism becomes the dominant pathway for its breakdown. The resulting gas production reflects this shift and contributes to changes in the physical and functional state of the intestine. Microbiome shaped by digestion, not vice versa. The microbiome reflects the output of digestion rather than acting as the primary driver of digestive function. Microbial populations adapt to the substrates that reach them, and those substrates are determined by how effectively upstream processes reduce and absorb nutrients. When digestion proceeds in sequence, most macronutrients are processed and absorbed before they enter regions of dense microbial activity. The remaining substrate is limited and consistent, and microbial behavior stabilizes around that input. When digestion is incomplete, larger amounts of partially processed material move downstream, expanding the substrate pool and shifting microbial activity. The type and quantity of substrate define microbial composition. Bacteria metabolize what is available in the lumen, not what is consumed at the level of the diet. When proteins are fully reduced and absorbed, minimal protein substrate reaches the colon. When this reduction is incomplete, larger fragments persist and become available for microbial metabolism. The same pattern applies to carbohydrates and fats. Increased delivery of fermentable substrates drives changes in microbial activity, which then alters the chemical and physical environment of the intestine. These changes in microbial behavior are secondary to the conditions established earlier in the digestive sequence. Fermentation, gas production, and the generation of metabolic byproducts increase when substrate availability rises. These outputs influence motility, luminal pressure, and epithelial interaction, but they do not originate independently. They reflect the presence of substrates that were not processed upstream. The microbiome responds to these conditions, amplifying their effects within the intestine. The location of microbial activity also shifts with changes in digestion. When upstream processing is effective, most substrates are absorbed before reaching distal regions, and microbial metabolism remains localized. When digestion is impaired, substrates appear earlier and in greater amounts, moving microbial activity proximally. This alters the pattern of fermentation and increases interaction between microbial byproducts and the upper sections of the intestine. Microbial populations adapt over time to the substrate patterns they encounter. A stable digestive sequence produces a stable microbial environment. Variability in digestion produces variability in substrate delivery, leading to shifts in microbial composition and behavior. These shifts are not independent drivers but reflections of changing conditions within the lumen. Understanding the microbiome as a downstream output clarifies the relationship between digestion and microbial activity. Interventions that focus solely on altering microbial populations without addressing upstream digestion do not change the underlying substrate conditions that drive microbial behavior. Restoring digestive sequence reduces the delivery of unprocessed material, stabilizing the environment in which microbes operate and allowing microbial patterns to normalize in response to consistent input.